to all of you. Today we are going to perform a virtual lab on the topic study of controlled pollination. Okay? Controlled pollination. See, there is a symbol that is pollination, which going to be takes place with the help of different types of pollinating agent. What is pollination? It is the transfer of pollen grain from the anther to the stigma. Okay, first thing. Second, in this case I mean to say normal pollination, both female reproductive organ and anther need to be mature. Okay, then only it is going to be successful. Okay, that is the general thing. But what we are going to study here today is study of controlled pollination. What do you mean by controlled pollination? It means that in such kind of pollination that is not going to be takes place naturally. Okay, this is going to be performed according to our desire. According to our desire. So what are the topics and things that we are going to be discussed here? Same as in learning objectives. Why we are doing this experiment? What we will learn? See, this is a practical experiment. Practical. It means that this method we can use in our life for our betterment. Got it? And then material required, what is the theory behind that, and instruction and demo. Okay. So what is in to perform emasculation, bagging, tagging for controlled pollination. I told you desired pollination, controlled pollination, and that will be achieved only by the method of emasculation. Because what I told you, pollination is the method of transfer of pollen grain from anther to stigma. So, if we remove the anther, there will be no pollination because no pollen grain going to be fall. So, in that case, what we will do, in case of emasculation, we remove the anther and we dust the pollen grain of our choice on the stigma. Okay? Next is bagging. Why that is necessary? It is very much necessary that we should cover that emasculated flower so that unrequired, undesired other pollen grains should not get fall on the stigma. Then tagging. It is very much necessary that we put a tag on that, on which day, which species of plant we have choose. Okay. If there is some special characters, that all those things can be going to be tagged there. Okay. Then what is the objective? See, perform emasculation, bagging and tagging for controlled pollination. Handle the specimen and apparatus carefully by following all the precautions given in the modules. Minimize the errors and mistakes committed during the experiment. See, one more thing. In any subject, practical subject, you will see there will be a topic that is known as error. Okay. Practical hand. Everyone have not the perfect practical hand. Practical hand is also a skill. It is not that much easy or anyone can learn. It is very necessary that how you are performing the experiment, how you are handling the instrument. There is a protocol, there is a system to handle the instrument or different kinds of materials that we use in a practical lab. So, and for that, it is very much necessary that you have a proper observation. Until unless you won't have the proper observation, you are not going to be master in the case of practice. So it is very necessary to observe the things very properly during demonstration. Okay? So learning objective that once we learn this, the percentage of error of performing the control pollination going to be minimized. Okay? And all this method you can use for your, I need to say economical purpose and such kind of things. Okay? Materials required. A bisexual plant, simple. A bisexual plant because emasculation is a method in which we are going to remove the male gamete, that is androecium. Okay? So bisexual plant, mature flower. Flower needs to be mature. Different varieties of pea plant and different samples we can use. Hand lens so that we watch them carefully and forceps to hold, to separate and for such kind of things. What is theory behind emasculation? See, nowadays this plant breeding, nursery, horticulture, things are in fashion. Okay, because we all know that population is getting increased, land is getting reduced. Okay, so there is also a need of advanced methods, successful methods. So all these methods are going to play a very much important role in upcoming future. So plant breeding is the science of improving and producing new plant varieties. New plant varieties. Conventional plant breeding programs involve crossing different species to produce commercially superior variety. And by this method, we can develop a superior qualitative varieties and products. Okay, that is why it is necessary. Okay. Are you seeing all these things? See, just have a look here. Okay, carefully. Wild mustard plant. We all know mustard plant. Okay? Wild mustard plant, and from that mustard plant, by doing such kind of controlled pollination, hybridization, what we have evolved. Just have a look. Kohlrabi. Okay, in local language that is known as plant kohlrabi and such kind of things. Okay? Then you kale, leafy vegetables which is used for the purpose of salad and sandwich, which is highly proteinaceous. Broccoli. By doing such kind of methods in agriculture, we evolve so much things. Got it? So this is the advancement of using these methods. Now, what we have did in such case? Strain that we sample we have used kohlrabi. What is the modification stem? Here modification of leaf. Here flower buds modified, and that is green in color. Here lateral leaf buds, the leaf which arise laterally at the nodes, they are condensed in the form of bristles, in the form of sprouts. Then terminal leaf buds. In the case of cabbage, the terminal end of the leaf going to be modified, okay, as a storage form. Then here flower buds. So in that particular region we have made the experiment. In controlled pollination, now come to the main topic. In controlled pollination, male and female parents having desired trait are crossed. But consider desired trait that we need, the trait, the character that we want in our plant. Artificial hybridization is one of the major approach for crop improvement. Major approach for the crop improvement program. See how emasculation can perform either with the help of tweezer or with the help of scissors or set. What we do, remove stamen from purple flower. There is a flower from which the stamen, that is male reproductive organ, get removed. Second, transfer sperm bearing pollen from stamen of white flower to egg bearing carpel of purple flower. It means that in the case of purple flower, we remove the male reproductive organ. What present here is the female reproductive organ. And from here, from the white color flower, we have choose the pollen grain. We have choose the pollen grain, and we dusted that pollen grain on the female reproductive organ or the stigma of the purple flower. Got it? This pollination we made between the purple and white color flowers. Now, pollinated carpel mature into pod. This carpel going to be matured into the pod and planted seed having the round seeds. We got round and wrinkle seed, right? In case of round seed, the content of starch and nutrients is more and higher as compared to wrinkle. So this is what we got here, the advantage of doing such kind of controlled pollination. Then after that, we examine offspring all purple flowers. We have made the cross, we sowed that seed again, and what we will get, all the flowers are purple in colors. This is achieved by the process of emasculation, where the stamen are removed from the bisexual flower of the female parents, okay, in order to avoid self-pollination in this flower. It is done before the anther gets mature. once anther gets mature, it is going to be rupture and pollen going to be fall. So it is very much necessary that we remove the anther before maturation, because if it is going to be mature, it is going to be dispersed or fall on the stem, and our aim going to be failed. Okay. This is achieved by process of emasculation, where the stamen is removed from bisexual flower female parents in order to avoid self-pollination in this flower. It is done before anther get mature. Okay. See, this is how we are going to be perform the experiment. Anther is going to be removed.
So, previously you have told us that the flower must be mature.
For the pollination, flower needs to be mature, but we need to be in the observation condition, because if that anther is going to be mature, then it is going to be ruptured. So before dehiscence of the anther, it is necessary to remove that. What I told you, in general condition, both need to be mature. Then only pollination going to be successful. But in artificial condition, in controlled pollination, it is necessary that we remove the anther before maturation. Okay. So the stigma is protected from contaminating unwanted pollen by the process of bagging. Next step is bagging, so that unwanted pollen grains should not cross or should not fall on the stigma. Okay, and that is the reason why we bag the emasculated flower. Okay. The flowers are repacked and the fruits are allowed to develop. This is what we are going to be performed. This is artificial pollination. This method is employed in plants having large flower. Those who are having large flower, there we can achieve successful pollination or fertilization by using this kind of methods. Okay. Here is next is your instruction. And the same instruction will be there in the demo. So have a look here, the demo.
Emasculation of flower.